There's tremendous complexity happening all around us in nature, but essentially it's invisible to us. It's happening at the nanoscale, at the molecular level. And so what my lab is trying to do is build new technologies that enable us to see biology in its full complexity and beauty. Advances in genomic technology development have really driven our understanding of biology at the level of the gene, at the level of DNA. But where the rubber really meets the road is at the level of proteins. These are the molecules that genes actually code for. So current protein sequencing and analysis technologies, how they work is they're actually fragmenting the protein up into small fragments, and then they're computationally stitched back together. And you end up losing a lot of information about what was the actual state of a full-length protein molecule. So what my technology is trying to do is read individual protein molecules end to end in the natural way that they exist within the cell, to really understand how each protein functions at the molecular level. That would have applications in understanding just fundamental biology, but also understanding how, when biology goes wrong, how could we engineer new diagnostics so that we can detect it sooner, as well as new therapeutics, how can we ultimately treat it. Our research group is the Molecular Information Systems Lab. It sort of sits at the interface between computer science and biotechnology. And so as part of that group, I get to work with a really eclectic group of students and scientists and other researchers spanning molecular biology to biochemistry, microbiology, to experts in machine learning, software engineering, and hardware. We're constantly sort of learning from each other and being able to contribute to the overall directions that all these different projects are going. We actually have a full molecular biology wet lab here in the computer science building. And I really don't know of any other computer science schools that have this. We've established some proof of principle experiments that we're able to actually read individual protein molecules end to end, full-length protein molecules. And so what we're really excited about, let's say in the next 5 to 10 years, is seeing this technology actually translating out of our lab and into the hands of real users, other biologists, clinicians, engineers, so they can start to understand biology now from a more fundamental level. I'm Jeff Nivala, an assistant professor in the Paul G. Allen School of Computer Science and Engineering.